Good afternoon. And coming up as the last speaker on a packed day like this is a challenge in itself. So continuing on the wings of what Dr. Tai-Ping has been telling you, traditional medicine has for ages been prompting us to pursue that so that it becomes a way of maintaining health and also preventing diseases. And as Dr. Tai-Ping told you, natural products over the past 200 years has been the source of medications, of effective drugs. In fact, about 50% of all blockbuster drugs that are available in the market today have their origins in natural sources. And what this has enabled us to do is take these natural products and apply them and facilitate experiential knowledge. So novel drug discovery, whether it is from natural sources or conventional drug discovery approaches, requires a lot of intense effort. It takes close to $1 billion to develop a new drug, and it requires a lot of experimentation, a lot of intense research. And that is true for natural products as well. More recently, newer approaches, newer technologies, high throughput screening, where hundreds of thousands of source molecules can be screened to identify potential lead molecules, or high content screening, where you're looking at multiple parameters at the same time and identifying and analyzing and testing and complying with the requirements, the guidelines of the US FDA or other regulatory authorities. And looking at complex mixtures that these natural product extracts are becomes a challenge, because you're looking at multiple interactions, you're looking at a large number of targets, and then looking at multiple metabolic byproducts and being able to identify what is the active ingredient in this complex extract becomes a challenge in itself. So that's where we decided to take a different approach. And as Manisha and others have told you, our approach to science comes with directions from our Chancellor Amma. So in this case, while in an interaction with her, Amma asked me, what do you do in terms of research? And when I explained to her that one of the problems that we were contemplating solutions to is, why is it that in case of diabetic individuals, there is a delay in the healing of wounds? And she said, the answer exists in traditional medicine. People in the villages, and she herself has experienced that, people in the villages around in India have used the outer shell of the cashew nut. They heat it up, there's an oil that comes out of that, and you apply that oil to the wounds, and the wounds are healed. Said, go find out why. And that's what we did. We went to the India and Kerala, where we are located, is the cashew capital of the world. We went out to the cashew factory, brought back a bunch of cashew nut shells, and processed it through the lab and extracted a compound called anacardic acid, which has a structure like that, and also isolated a lot of other compounds, and then turned our focus to a group of enzymes or proteins that are very important in this wound healing process. And that's where I have had the benefit of extensive mentoring from Professor Gillian Murphy, at the University of Cambridge and Professor Hideaki Nagase from Oxford University, who are absolute authorities in this area. And we started looking at this group of enzymes, and I won't go into too many technical details, but suffice to say that these enzymes, or these proteins called matrix metalloproteinases, are involved in a number of pathophysiological conditions. You take neurodegeneration, you take arthritis, you look at least 15 different forms of cancer, you look at myocardial infarction and delayed wound healing. All of these group of enzymes, the matrix metalloproteinases, have been implicated in. And what we did is went in and isolated that compound anacardic acid and then tested it in cells and found that, surprisingly, this compound anacardic acid brought about a very strong dose-dependent inhibition of the activity of these matrix metalloproteinases, or gelatinases as they are called, MMP2 and 9. Nice dose-dependent inhibition. And then in collaboration with our friend at Scripps, Jeff Perry and John Tainer, we were able to show that in some elegant docking studies that Jeff and his group did at Scripps, this compound actually docks right into the active site of that enzyme. And the carboxyl group binds zinc at the active site, and the aliphatic side chain, this long aliphatic side chain, actually inserts itself into what's known as an S1 pocket in that protein and probably locks up that protein in that conformation, which made it very interesting because the carboxyl group, this aliphatic side chain, and the hydroxyl group seem to be involved in this inhibitory activity. Because when we looked at other compounds which are common drugs, salicylic acid or methyl salicylate aspirin, share a similar structure with that anacardic acid, except that they lack that side chain or they lack the carboxyl group, and they have absolutely no inhibitory activity of this enzyme. We went on to synthesize some other molecules that have similar structural capabilities, and one of those was biacacetin. We found again strong inhibition of this gelatin degradation, strong inhibition of the migratory properties of these cells, and what was even more interesting is that in a combination study of anacardic acid with this biacacetin, suddenly anacardic acid that was only inhibitory at micromolar concentrations suddenly became potent at nanomolar concentrations, giving us the potential of a hybrid molecule and possibilities of embarking upon that kind of activity. So we're going on to try and synthesize other derivatives that share structural similarity with this anacardic acid. What I want to point out is that where conventional drug discovery looks at sources that can be very expensive, here you're utilizing waste and trying to develop drugs that could have potential applications. I'd like to switch channels here and look at another aspect, another story that we have been working on, which is wastewater. Water, we know, is an essential, vital, critical resource, but managing the waste water has become a monumental challenge. Out of sight, out of mind. We really don't look at these statistics, but they can be alarming. Very few of us think of the fact that 2 million tons of sewage is discharged into the world's waterways annually. We really don't pay much attention to the fact that 1.8 million children below the age of 5 die annually because of water-related diseases, which in other words is 1 in every 20 seconds. We don't pay much attention to the fact that 90% of all wastewater that is actually being discharged especially in developing nations, is not treated at all. So that is polluting the waterways, providing contamination and challenges in dealing with that. So it's no more a luxury. It is what you can call a prudent, practical, and transformative action that needs to be taken that has implications in public health, has implications for sustainability and employability. That just gives you an idea of what the picture would be like for urban cities with 100,000 population by the year 2050, and India, China, and even the US figure high on that list, which is where the source of contamination of these waters. So essentially what we are talking about is that there is a lot of wastewater that's collected. There is 165 billion metric cubic of wastewater that is collected every year. But how much of that is actually used? Barely 2% is actually reused. Half of the developing world today does not have access to adequate sanitation. And also, there are close to 900 million people who do not have access to safe drinking water. That is the challenge. And what essentially needs to happen, what is the need of the hour, is innovation to reduce the volume and the contamination of the wastewater that is being generated. Look at methods to treat and recycle this wastewater and find affordable solutions for that. You heard in the morning about Bhavani and her group at Amrita University building toilets. We have now embarked upon a strategy to try and treat the wastewater. And the joke around now is that with the toilets being built and the wastewater being treated, our future is in the toilet. So essentially what we are trying to do is look at several different ways where this wastewater can be effectively treated. And with adequate funding received from the Bill and Melinda Gates Foundation earlier last year, along with the Department of Biotechnology of the Government of India, BIRAC, we have set about addressing this target. And what we are looking at is a combination of bacterial viruses or bacteriophages, which are very specific in infecting their specific hosts, and eliminating those bacterial pathogens. Also looking at enzymes as well as bacteriocins and small molecules that we have experience in isolating from natural sources, and creating what we have called a Lytics Broadcasting System, where a mixture of these agents can actually help us to reduce infection by bacteria, for example, Vibrio cholerae or E. coli or Shigella, and also addressing the parasite problem, Giardia, Ascaris, and so on, as well as odor-causing bacteria. Those are some of the major problems where sewage treatment is concerned. So we have, and I won't go into too many technical details, but we have taken each of these cases and looked at how to solve the problem, and have had considerable success in practically eliminating bacterial contamination as well as viral contamination. Then one other aspect of what I want to touch upon, India also has the dubious distinction of being the diabetes capital of the world. We have close to 65 million Indians who are diabetic, and World Health Organization projections are that by the year 2035, that number may have crossed the 100 million mark. And so that in itself is a big socio-cultural problem, and what we have started to do in collaboration with Wipro Technologies, and our collaborator Arpita Banerjee is here, we have developed two devices an automated insulin pump for which we have a US patent, and a non-enzymatic glucose sensing system. In collaboration with Wipro, we are now enabling those devices to be Bluetooth capable, have the data from there transferred to a mobile application which Wipro has developed, and the data would be transferred to a cloud, hosted on a server, and then act as a link between the diabetic patient and a diabetologist, a endocrinologist, a diabetes helpline, and so on, and in that way be able to provide better diabetic health management for the patient population. So the take-home message that we would like to leave with you is that these are solutions that need to be developed and evolved over time, and that is of paramount importance. But these have to meet the requirements of socio-cultural sustainability. They have to be compatible with technological and institutional feasibility. They also have to be cognizant of economical affordability and environmental acceptability. Those are collective efforts that have to be sustained, and those are the needs of the hour. Thank you very much.